Respiration is getting energy from breaking down our food molecules to shove that third phosphate back on. So that's how we use and make ATP, but it takes a lot of work to make it. Cellular respiration comes down to three processes. Glycolysis, which is going to just break down glucose. You can tell that from its name. Glyco is glucose, lysis. We get a little bit of ATP out of that. Second is the citric acid cycle. This is going to break down the fuel the rest of the way, so there's nothing left of it but carbon dioxide. And that's where our carbon dioxide we're always exhaling comes from. And then the electron transport chain is where we're going to make most of the ATP. You can probably guess something that it does from its name.
Transports electrons.
Yep, transports electrons. Those electrons also come from the food molecules. So, glycolysis cuts up glucose. What goes in is a glucose and 2 ATP. That sounds kind of weird because our goal is to get ATP, but we'll explain why that happens coming up. What comes out of it is 2 molecules called pyruvates, 4 molecules of ATP, so we put in 2, we get 4, and some electrons and hydrogen ions. Glycolysis happens in the cytoplasm, so you don't need mitochondria for this. In fact, red blood cells don't have mitochondria, so they get all of their energy from glycolysis. So it's 10 chemical reactions, but we're going to break it down into 3 general steps. First, spend 2 ATP to get it started. Those phosphates get attached to the glucose. Now, this is often referred to as priming the pump. I think of it as, anybody have a gas-powered lawnmower where you have to yank on that cord to get it started? Okay. So, spending the ATP is yanking on the cord, putting some energy in. And then once the motor is running, it uses its gas, here it's using glucose, to get more energy back out. After we spend the ATP, then we cut the glucose in half. So we get 2 pieces that are 3 carbons each. The rest of glycolysis involves making some adjustments to those 3 carbon pieces until we end up with the pyruvate or pyruvic acid. You'll see it written both ways. Technically, the difference is whether the hydrogen is attached to it or not. If it's released, then it's pyruvate. As you're doing those reactions, that's where you get the 4 ATP and some electrons and hydrogens. Now, those are shown here as NADH. I'll talk about that next. What I want to do first is come over here and sketch out glycolysis. Put in the 2 ATP and the glucose. And spend the ATP, cut the glucose, and turn the pieces into pyruvate. And so out of that, we get 4 ATP. Those we're done with, the cell can use them. We get 2 pyruvate and some electrons, which I'm going to write as E-, and hydrogen ions. So that's glycolysis in a nutshell. or in a cell. NADH is a molecule that fits into a category called electron carriers. So electrons out loose are electricity. Hydrogens out loose, what are they? Or let's say, what kind of electrolyte? It's the reason the test is called pH. Acid. Okay. We don't want electricity and acid wandering around in our cells. So the NADH grabs them. So far, we've spent 2 ATP and produced 4. So we have a net gain of 2 ATP from each glucose that goes in. It's not great. We can do better. We're going to do better with those electrons and hydrogens in the electron transport chain. Questions on glycolysis? Okay. Where's the sanitizer? Thanks. Right, next is the citric acid cycle. The purpose of the citric acid cycle is to collect a lot more electrons from our fuel molecules. The 2-pyruvate from glycolysis go in. What comes out is 6 carbon dioxide, 1 for each carbon from the glucose, 2 more ATP, and some more electrons and hydrogen ions. Citric acid cycle is going to take place in the matrix of the mitochondria. If you remember mitochondria structure, we got outer membrane, inner membrane. Inside that is the matrix. We're going to call this area the intermembrane space. Okay. So what happens in the citric acid cycle? First, we're going to break a carbon off the pyruvate, turn it into something called acetyl-CoA. CoA stands for coenzyme A. That is, starts out as part of the enzyme and it gets attached to the other 2 carbons. It's going to come back off later. For simplicity, we'll just think of acetyl-CoA as a 2-carbon molecule. Okay. So 3 carbons of pyruvate go into acetyl-CoA and CO2. Then steps 2 and 3 are actually happening at the same time through a sequence of 8 reactions. The acetyl-CoA turns into 2 more CO2. So for the glucose, 2 pyruvate went in, we get 3 CO2 from each pyruvate, so 6 CO2. And while we're doing that, we produce 1 ATP for each pyruvate that goes in, so 2 per glucose, and lots of electrons and hydrogens. They're carried by both NADH and FADH2, which is another electron carrier. If you're interested, I can tell you what NAD and FAD stand for. Short form, N is derived from niacin, vitamin B3. F is derived from riboflavin, vitamin B2. They're fairly large molecules, and they have places where they can hold these extra electrons and hydrogens. This is what the citric acid cycle looks like. The arrow here is turning the pyruvate into acetyl-CoA. And then this cycle, the first thing we do with the acetyl-CoA is attach it to another molecule and make citric acid. And that's why it's called the citric acid cycle. as we go through, we take the carbons off, we collect our electrons in 4 different places, we make an ATP. We end up back with the same molecule that joined with the acetyl-CoA in the first place. And that's why it's a cycle, is we add an acetyl-CoA and then we come back around to where we started. All right, before we go to the electron transport chain, citric acid cycle, the pyruvate goes in. Step 1, we turn it into acetyl-CoA plus CO2. Step 2, turn acetyl-CoA into 2 more CO2. Collect electrons and hydrogens. So what comes out is 6 CO2. And we don't want that, we just exhale it. 2 ATP, we're going with 1 glucose's worth. And more electrons and hydrogens. Questions on citric acid cycle? Finally, the electron transport chain. It's where we're going to make the largest amount of ATP. We put in the electrons and hydrogens from the previous 2 stages. We also need to put in some oxygen. What we get out is water and ATP. So technically, we're also putting in ADP anytime that we're getting out ATP, but it's simpler to just skip over that. The electron transport chain happens along the inner membrane of the mitochondria. We have a bunch of proteins that need to be lined up to transport the electrons. So having that membrane lets them be held together in the right order. What happens in the electron transport chain? First, electron transport. It's kind of like a game of hot potato, where these proteins on the membrane take the electrons from NADH and FADH2, and they pass them along, and then oxygen gets stuck with them in the end. Now, you give electrons to oxygen, it becomes negative. If it's negative, it's going to attract something positive that's floating around. What positive thing did we have? Hydrogen. If we get 4 of them, then we're going to end up with hydrogen and oxygen makes water. So the water comes from the oxygen. The other hydrogens that aren't getting sucked into the oxygen are getting passed out into the intermembrane space, and that makes a gradient. A gradient is potential energy, like wind or water flowing downhill, and you just need the right device to capture that energy. We have the right device in our mitochondria. It's going to capture that energy to attach the last phosphate to the ADP. So, looks something like this, where purple is the proteins, yellow is where the electrons are being passed along. There's a typo in this diagram. It should say 1/2 O2 plus 2H equals H2O, or it becomes H2O. Then the red arrows are where the hydrogens are being sent out into the intermembrane space. Now, they're crowded over there. They want to get back in where they're less crowded. There's only one place that they're allowed through, and that is a protein called ATP synthase. ATP synthase is an enzyme that uses the energy from hydrogen flowing through it to jam that last phosphate onto ADP. And it's got a channel inside, and it's got kind of a wheel that the hydrogens turn as they go through, very much like a windmill. And that pushes the ADP and phosphate together.